Thank you. Sorry for the delay, but now we will start our session. And the first talk will be a lightning talk by Diego he was associate professor at the Université Paris, and he will present to us his MIToS evolution.
Okay. So good morning, everyone. Today I will present my package MIToS and the new features for the last release 3.0 in this week. So MIToS is actually a package for working with protein sequences and structures. So basically, proteins are the main machinery in the cell that performs all the functions that keeps all the organisms alive. And in particular, we can think of a protein as composed of a sequence of amino acid residues that determines a structure or a series of structures in the three-dimensional space that finally determines the function of the protein in the cell. So MIToS offers the MSI module that has ways to represent protein sequences and multiple sequence alignments. The PTV module for working with protein structures, and the and the information module for working with evolutionary information as conservation and covariation can give us a hint about the function of a protein. So in the last year, revolution has happened because of AlphaFold 2 in structural bioinformatics, because now we can predict very accurate structure for proteins using simply multiple sequence alignments. So that has allowed us to go from 200,000 proteins in the PTV database to 200 million proteins in the AlphaFold DB. So to access all this information from MIToS, now we have two functions. We have the download AlphaFold structure function that takes an uniprot accession that identifies the protein and downloads the structure for that protein from the AlphaFold DB. And also we have a carry AlphaFold DB function that is going to perform a carry to the AlphaFold DB API, and it's going to return a JSON object that we can use as a dictionary to get more information. So for example, here, if we want to see what is the quality of the model, we can try to see the PA matrix, and we can get the URL from that. So also for integrating MIToS in pipelines that use AlphaFold 2, we have added some new functionality to the MSI module. So one of the functionality is that now it parses read and write in ITM formats. That is the format that is used by AlphaFold 2 and CollabFold. So now you can use it, for example, to analyze the multiple sequence alignment that AlphaFold has used for predicting the protein structure. And we also have, in order to test different things about and to generate variability, for example, in the structural models, a new shuffle MSI function that keeps the reference sequence untouched, but allows you to, for example, shuffle only a subset of columns of the multiple sequence alignment. And also very important when trying to predict protein-protein interactions, we usually create concatenated alignments in which we match in the same row sequences that came from the same species. And in order to make easy this process, we have now the join MSA function that is going to take the two multiple sequence alignment and a list of pairs of a dictionary indicating the pairing of the sequences in the multiple sequence alignments. And it's going to create these concatenated alignments for AlphaFold 2. So now also we have BioStructures support in MIToS. Thanks to now BioStructure is very lightweight, thanks to show. Thanks. So MIToS now has a dependency BioStructure that allows to, for example, read and write MNC file that wasn't possible before. Now it is also the default format for MIToS, so when you download, for example, the structure from AlphaFold DB, it's going to be downloaded in the MMC format. And And another thing we add is a way to convert between the vector residue, PDB residues of MIToS to the molecular structure of BioStructure from BioJulia. So that allows to use to go between the two structures in order to expand the utilities to use other packages of the BioJulia ecosystem. So, for example, here we have an example of going from a MIToS structure to a molecular structure of BioStructure, and then using the extension of BioStructure for data frames that converts the structure to a data frame object. So if you are used to work with with the structures as a data frame, you can use that. So for concluding, MIToS 3.0 has many functionality to work with the state-of-the-art tools. Here in the talk I was mainly talking about AlphaFold 2, but also it integrates things to work with FoldSeq. And in the future I would like to expand the integration of MIToS with the BioJulia ecosystem, and also with the available GLL packages for external bioinformatics tools, as for example MAFT. So thank you all for your attention. I will be glad to answer any question you may have. Yes.
I'm not familiar with MIToS. Does it also include functions for... Thank you. Does it also have functionality for plotting structures, visualizing structures, and working with structures visually?
So for visualizing structures, there are a very basic plot recipe that you can simply use plot and plot the structure or the multiple sequence alignment, and it's going to show a very basic representation. However, in the ecosystem, there is something that is better, that is Biomaki, that allows to represent the structures in a more beautiful way and interactive way, and also the multiple sequence alignment. So, but you can use it also with MIToS.
Thank you.
Thanks, great talk. You mentioned FoldSeq. How does that work? Do you use some binary or something to run FoldSeq?
Yes, FoldSeq is already available as a GLL package, so you can install FoldSeq GLL to install it and run it from that. Basically, the new things in MIToS for working with FoldSeq is mainly the ability to parse the PDB structures that FoldSeq generates. For example, when you carrying the AlphaFold DB, this is a very large database. It only stores the alpha carbons, and the PDB format it outputs is not very compliant. So now it can parse also this output, but in fact you use it as an external tool. Okay. So thank you very much for your questions.
Yeah, let's thank the speaker again. And Vincent, you can come to the stage.